Yes, good afternoon, students. Today I am going to discuss about mm, DNA structure. Actually, DNA. Before that, I can explain. Mm, this is a common DNA. This is a common genetical material in the cell. Usually, whatever microorganisms like bacteria and virus and even human beings which are having the genetical matter in the nucleus. um, in the nucleus, the genetical material which is called as DNA. See, um, the structure of the DNA is double helix basically, and which contain 10 nucleotides. See, each spiral strand composed of the sugar phosphate backbone and attached to the basis is connected to the complementary strands by hydrogen bonds. And the DNA which are having fluids and pyramids like adenine and guanine, thymine and cytosine and guanine. See, these are the common structure. Same, the DNA structure, the first discovered by the scientist name, the scientist that is belongs to Watson and Crick. See, the Watson Crick postulated a three-dimensional model of DNA structure that accounted for the both the X-ray data and the characteristic base pairing in the DNA. See, it consists two helix, which means which are having two standards, and polynucleotide chain. The polynucleotide chain coil around the same axis form a right-handed double helix. See, in the DNA which are having two ends, 3 to 5 and 5 to 3. These are the most important two strands which are present in the DNA. And the backbone of each chain, which consists alternate phosphate sugar, sugar phosphate, and also in two DNA helix which is bind with hydrogen bonds, double bond and triple bond. Now I can explain in the DNA which are having purines and pyramids. See, for example, purines. I'm going to discuss about the purines, and purines again classified into mainly two types, that is adenine and guanine. And pyramids is also classified into mainly two types, that is nothing but thymine and cytosine. See, the adenine in the first strand of the adenine which is bind with thymine. At the same time, the guanine bind with cytosine. See, A bind with T with double hydrogen bonds, and also G bind with and guanine bind with cytosine with triple bond. See, and in the base pair are 0.34 nano m, means nanometers, a part in the DNA helix. See, a complete turn of helix takes 3.4 nm. Therefore, in each helix which contain 10 bases, the external diameter of helix is 2 nm only. Right now, the helix has two external grooves. The narrow groove is called as minor groove, while the wide groove is called as major groove. The major groove is the site of the DNA binding proteins. The major grooves often are the site for binding small molecules. See, it's a very important here, the purines and pyrimidines. The pairs of bases are always between purine and pyrimid, especially the pair A bind with 2 T and G bind with the C. See, in the between the A and T which are having two hydrogen bonds, especially in the between guanine and cytosine which are having triple hydrogen bonds. See, can you see here that two chains do not have the same base compositions? I mean it indirectly, these are the non-identical but two chains are complementary to each other. Such chains are called as complementary chain. For example, the first chain which contain A adenine, thymine, A adenine, and cytosine, Z guanine, C cytosine, A adenosine, and cytosine. Next, and 3 adenosine, I, 1 thymine, 3 cytosine, and 1 Z. See, the opposite pair chain, which means the second chain which having in the thymine. Why? Because in the first chain which are having the first nucleotide, first nitrogen base is A bind with T is nothing but which in the second chain which started initial, that is A. These are the common binding A with T and G with C. Usually in the pyramids which are having one more nitrogen basis is present, that is nothing but uracil. So very important, uracil which is present in the RNA structure, not in the DNA. In the RNA, uracil, which is, I mean it in the RNA, the thymine is completely absent. The thymine replaced with uracil, even though the adenosine bind with uracil in the structure of RNA with a double bond, right? And now I can explain. I can explain the structure of the DNA. In the structure of the DNA, what is main difference between thymine and in the structure of RNA uracil? See, in the structure of thymine, which are having majorly 6 carbon acid. See, you can see the structure of pyramids and sugars is usually see that in the thymine, that in the position of the third carbon which are having methyl group, in the uracil at the position of the third carbon which are having I am extremely sorry, and in the thymine at the position of the third carbon which are having CHO group, means alkali group, and also in the thymine which are having methane group. That is the main difference between thymine and uracil. The thymine which is present only RNA structure. See, these are the this is the common DNA structure which are having main three components, that is nothing but deoxyribose, usually the ribose sugar, either pentose sugar which is also called as, and base, four different nodes ones, and phosphate. I mean bases, I mean nitrogen bases usually, which are having four different type of adenine, guanine, cytosine, and also cytosine, and also adenine, thymine, guanine, and cytosine. See, this is commonly the structure of the DNA which is called as nucleotide. Nucleotide. See, you can observe the nucleotide is meaning nucleotide, which means which contain tide, which means which contain how many parts? Usually three parts. Tide, which contain three parts, like deoxyribose sugar and nitrogen bases and phosphate. See, at the same time in RNA are also having only only two components, that is ribose sugar is present and nitrogen bases is a similar character with the DNA, and phosphate is completely absent. That is that type of RNA which is also called as nucleoside. So here DNA nucleotide and the RNA nucleoside, that is also one of the major differences between RNA and DNA. After that I can explain the DNA and RNA differences right now. Next, these are the common sugars, ribose and deoxyribose. See, which are showing similar characters. See, at the position of the carbon, third carbon, here showing that the carbon of the second, that is OH and H group. See, in RNA ribose sugars are present, in DNA deoxyribose sugars are very important for MSc. These are the main two sugars are present. See, next one, the bases, nitrogen bases are mainly two groups or two types or two components. These are the called as perinoids and purines. See, the perinoids made up of one six member ring, that is nothing but looking like a hexagonal shape. I can show the blue in colored, usually blue in colored. Next one, the purines with made of six member ring fused to a five member ring, and the pyramids are which are classified only two, that is thymine and cytosine. Even purines which are having adenine guanine. See, the rings are not only made of the carbon, specific formulas and the structures are not required for IB. Next, even here the purines which are having one more type, that is uracil, which are present in the RNA. See, now the what is the functions of the DNA is nothing but see, no, just wait, functions, after that I can explain the functions. See, you can observe the DNA double helix structure. See, the DNA double helix structures which are present, three different type of the components. See, in the corners you can see the P, that is phosphates. In the DNA we can see in the phosphate. You can observe in the diagram in the middle position which are having nitrogen bases. See which type of nitrogen bases, adenine bind with T and cytosine bind with Z, and guanine side bind with C. Even that thymine bind with A adenine, and C, cytosine bind with guanine, and finally that is guanine bind with cytosine. See, these are the nitrogen bases which are present in the central side, and also phosphate group is also present in the nitro, and sugars are also present. Next, which are showing two helix, I can show the diagram to the 5 dash to 3 dash and 3 dash to 5 dash. These are the main DNA double helix structure in detail, right? So the sugars which is present in the outside, these are the backbone, connect with the nucleotides in the chain. See, hydrogen bonds between the paired bases hold the two DNA strands together. DNA strands are anti-parallel. This is also very important. You can observe the general structure of the DNA helix which are showing two strands. This is the first diagram, and each diagram one helix which are having 3.4 in name, and in one each DNA which are having nitrogen bases, each helix which contain 10 nitrogen bases are present. So very important. And each nitrogen bases in between two nitrogen bases which are having only 0.34 in name space. See, this is the very important first diagram. You can observe the hydrogen bonds, how to bind with thymine and adenine. Even guanine bind with cytosine, right? Then third one, the cytosine bind with guanine. Next one, even the final, that is adenosine bind with thymine. Yet then the general structure of the DNA, these are the common internal structure of the DNA and clear diagrams. Now I am explaining the DNA structures. See, now I can explain the different type of the DNA structures. We have majorly three different type of the DNA structures are present, that is aDNA and bDNA and zDNA. So aDNA is one of the many possible double helix structure of the DNA. It is most active along with other forms when comparing B and zDNA. Helix has left-handed sense, shorter, more compact helical structure. Even it occur only in dehydrated samples of DNA, such those used in crystallography experiments, especially for aDNA. This is the second DNA type, that is bDNA, most common DNA conformation in vivo. See, this is a common DNA and favorable conformation rate, high water concentration. This is also known as Watson and Crick model of DNA. Why? Because which are having two helix, and the two helix which are arranged in opposite manner, and two helix which are having 2 points, 3 dash to 5 dash and 5 dash to 3 dash. Next, the first identified in fiber at 92% relative humidity. This is the bDNA structure, and a very important DNA, that is zDNA. zDNA is one of the many possible double helix structure of the DNA. Helix has left-handed sense, and it is most active double helix structure. Can be formed in vivo given proper sequence and superhelical tension, but function remains obscure. See, the ZDNA was first discovered in our India. That is a specialized group, Rodley group. So this Rodley group was proposed the ZDNA, and also Shishishekaran group is also first found this type of DNA, that is zDNA. Next one, I can explain the functions of the DNA. Usually the DNA which are used as genetical material in the nucleus especially, and what can I mean, usually nucleus which are having the nuclear membrane, and also in the central side cytoplasm, in the cytoplasm which are having like like a fibrous structures. These are the called as chromosomes. See, on the chromosome you can observe like a ball like structures, that is called as chromomeres. In the chromomeres which are having genetical material, that is nothing but DNA. This is the one of the most important function of the DNA, which are used as genetical material in the each cell. See, nucleotides are precursors of the nucleic acids, deoxyribonucleic acid, and also ribonucleic acid. See, and the nucleic acids are concerned with the storage and transfer of the genetical information, which are helping in the genetical, the transformation of the genetical material from one generation to another, even one plant to another plant. This is a very important in the DNA. And in the DNA which are having a specialized function, that is nothing but auto ketolysis. See, what is autoketolysis is nothing but that is a self formation of the DNA. I mean, the mother DNA which is produced two daughter DNAs, that is autoketolysis. Next one, and DNA must be stable. There are coding and non-coding regions found under DNA. Coding is non-coding is also very important. The code, see, codons is different. Why? Because the codons is not, the codons are related with the nitrogen bases, especially code which means we have triple codon, triple which means which are having how many nitrogen bases? Majorly three nitrogen bases, like adenine, guanine, and thymine. Sorry, for example, triple, I mean, three nitrogen pairs, which is also called as triple codon. Initial codon I can explain, that is AUZ. AUZ means adenine and uracil and Z-guanine. This is the initial codon which is influenced this particular amino acid, that is nothing but methionine. The first initial codon is a AUZ. This type of DNA nitrogen bases which are helping in the formation of the triple codon, even the triple codons and also nitrogen bases which are influenced in the protein synthesis process. So very important. The coding region code of genes, which is proteins. Non-coding regions can be either DNA junk or help regulate protein synthesis. See, sense codon and non-sense which are having codons two type majorly, that is sense codon and non-sense codons. See, for example, um, nonsense codons. I can explain the nonsense codons majorly. Nonsense codons are mainly three types. That is nothing but UAZ and UGA and UUU. See, these are the main three codons which are not identified, which is not related with any amino acids, hence which is called as nonsense codons. But the remaining, we have 364 codons are present in our botany usually, and the 64 codons, 61 codons are sense codons, which means these are the all 61 codons are influenced a particular amino acid, but only three codons are not it influenced any type of amino acids, then which is called as nonsense codons, right? These are the nonsense codons. Now, and the DNA which are helping in the and helping in the genetical material, the transformation of the genetical material to one to another area, sorry, one generation to another generation. Even the DNA which are helping in the fingerprinting. See, in the fingerprinting, you know very well about DNA fingerprinting process, which are helping in the crime reports, especially in the CBI and the Intelligence Bureau, IB, and so on, so extra. So the DNA fingerprinting is also very very important. Next, the DNA which are participating PCR test is nothing but one DNA which is formed so many so many DNA helix, which is called as PCR test, which are having chains, the DNAs which are showing like a chain, that is PCR test. The DNA which are helping in the PCR test, even this DNA is also which are helping in the biotechnology process of the cloning. So what is cloning? You know very well about cloning, I think so. The cloning method is now first the cloning method by the using of the genetical material of the DNA, using in the process of the cloning, which is form where we are taking a particular DNA from human whatever animal, then we are created at the similar character of the plant, whatever similar character of human being, by the using of the DNA in the process of cloning. That is cloning method. The cloning method is the cloning method first of all discovered in Tamil Nadu, in our India, I am telling, in Tamil Nadu state in the particular year, that is 1984, I think so. Then the cloning first they are tested in the animal, that is dolly, see, sheep, dolly sheep. Then after that, the cloning method, which is banned by the Supreme Court. Why? Because that is influenced and creating so many problems on the human beings especially. Then the Supreme Court will be banned after that, I think so 1990 maybe. So I mean indirectly the genetical material of the DNA which are helping all whatever molecular techniques and whatever biotechnology process. So not only that DNA printing, even cloning and PCR test, even which are helping in the protein synthesis process. And this is the genetical material which is influenced on all reactions of the cell, majorly I'm telling in this is influenced on all the reactions of the plants, whatever the reactions which is present in the plants, like photosynthesis and respiration, transpiration process in the human bodies, is also one DNA which are having a specialized function. If one DNA you can lose a gene, whatever a DNA you have, you have something uh, so you can see genetical diseases especially. So we have the chromosomes also there, that is chromosome structure. You can see this is the chromosome in the slide, which are having two arms outside, and you can observe the two arms to the lower side. In the arms in the red color, that is the DNA. So that I mean which are arranged in the spirally arranged, that is the chromatids. See, in the chromatids which are having a ball like structure, these are the chromomeres. In the chromomeres which are having this type of DNA genetical material. If in the we have For example, I'm telling human beings, we have how many chromosomes number is present in our human being is nothing but only 23 pairs up to 36 chromosome number. See, in this are again classified into mainly two types, that is autotrope and allotropes. See, autotrope is nothing but these are also called as vegetative chromosomes, which is not participating in the reproduction, and we have allosomes. These are the reproductive chromosomes, that is XX and XY. XX that belongs to female, and XY that is belongs to male. See here, if in the 46 chromosome, if one chromosome will lost now, then in our human body, which are showing one defect of in the human body. Hence hence the DNA is very very important for us. Whatever functions and whatever cell, even whatever process, then and next, then finally, last and but least, this is universal currency of energy, which means in the respiration process, which is form ATP energy. I mean, this is DNA which are influenced indirectly the formation of the ATP at the time of respiration. See, ATP means adenosine triphosphate is a nucleotide and derivative. Nucleotides are also components of important coenzymes like NADP plus, NAD plus and FAD, and metabolic regulators such as CAMP and CGMP. These are the most influenced on the respiration process, especially while what I'm telling, respiration process is nothing but the glucose, which means the carbohydrates. In the process of the photosynthesis, carbohydrates are formed. The carbohydrates which is converted into energy, ATP energy. These type of DNA which are helping indirectly in the synthesis of ATP. See, the synthesis of ATP is nothing but the reaction which is called as phosphorylation. What is the phosphorylation? So phosphorylation is nothing but the formation of ATP. Means ATP means ADP plus one phosphate which is giving ATP, which means ADP is nothing but adenosine diphosphate, which are having only two phosphate in the ADP, and also which is add a one more phosphate which is giving ATP, means adenosine triphosphate, which contain three phosphates. This is a very very important and so very important in the DNA. Euchromatin and heterochromatin. See, whatever in the chromosome structure, even DNA structure, we can see the chromatids based on the coloring. So if euchromatin and heterochromatin, both chromatins which are having colored, if colored less, that is hetero. If colored, I mean the chromatid which is observed more number of the color, that is belongs to euchromatin and less active, and heterochromatin is nothing but which are observed less colored and also very active in nature. These two is also very important functions of the DNA. This is common DNA structure and DNA functions and DNA internal structure and DNA structure, double helix structure. So thank you, and thank you one and all. Thank you.